Hi, I'm Amitha Karuppiah. I'm a rising junior at the University of Florida, and I worked in a Harrison lab this summer studying cell type marker gene expression in Metacoga truncatula. Yeah, so Metacoga truncatula is a model legume that my lab studies, and cell type marker genes, you can kind of think of them as cell name tags. So they're specifically expressed within certain cell types, and my project this summer was to make sure that the cell type marker genes were marking the cells that they were supposed to. So I did a protocol that would highlight the areas in blue where those cell type marker genes were expressed so I can kind of validate them to see that they were doing their job. And they were doing a good job, except for in these two cell type marker genes, they were found to mark primarily the vasculature, which is kind of like the veins of the plant, instead of the cortex, which is the layer under the surface of the plant. And I also looked at expression at two different time points, and then also in wild type plants and mutant plants. So this cell type marker gene marks AM colonized cortex cells, and arbuscular mycorrhizal or AM symbiosis is an important symbiosis where AM fungi transfer mineral nutrients to the plant in exchange for carbon compounds. So it's very important for things like phosphorus conservation and plant fertilization. And I found out that the staining was a little less intense in the mutant plants for all of these, including this cell type marker gene. So I did a root length colonization assay to see how much of the plant roots were colonized by AM fungi. And I found out that the mutant plants had less AM colonization than the wild type plants, which was pretty much consistent with the less intense staining that I had for this cell type marker gene. And then future directions that we might want to go with this is add AM colonization treatments to these cell type marker genes as well to see how AM colonization affects the expression of those cell type marker genes, and then also repeat this experiment to make sure the results are consistent.
Great, very, very nice. What was the most challenging part of this work, would you say?
The most challenging part of this work was probably taking these cross sections. I did them by hand, so I had to get very, very thin cross sections and get them in the right orientation so I could get good images.
Great. Well, it turned out very nicely. Thank you very much. Nice work.